Now to introduce today's speaker, Vladislav Kholostiakov. Vlad, I'm just passing the screen to you if you can accept the invitation to present.
Yes.
Now, Vlad
Hello.
recently completed his PhD with the Beyond Myrtle Rust program through the University of Auckland. He is passionate about microbes generally and applied this he's passionate about microbiomes generally and applied this passion to investigate the microbiomes of pōhutukawa trees, seedlings, and seeds for his PhD. We'll hear about part of that research today. Vlad, if you're all good there, feel free to get underway.
Thank you, Jenny. Kia ora koutou. Hello, everyone. It's a great honor to present some results of my PhD investigating seed microbiome of Metrosideros excelsa, New Zealand's most iconic tree, pōhutukawa, and its role in seedling development. It was a big PhD project under the supervision of Bruce Burns, Hayley Ridgway, and Mahajabin Padamsi. Before we move to pōhutukawa, I should remind you that previous webinar series focused on microbiome of Myrtle already found that some endophytic bacteria and fungi can really suppress myrtle rust infection. And they indicate that if we transmit microbial communities from resistant trees to susceptible trees, they can probably induce this resistance in very susceptible hosts. And we can think that if beneficial microorganisms could be vertically transmitted from parental trees to the seedlings, to their progeny, it would probably help to reduce inheritable resistance to Austropuccinia psidii, the pathogen which is causing myrtle rust disease. So, speaking about Metrosideros excelsa and myrtle rust, first, what we should think is why we observe these susceptibility levels, which are very different for different individual trees in urban environment and in some wild populations. We can see that some individual trees are highly susceptible to myrtle rust, while others, even being in close proximity to very heavily infected, demonstrate only mild symptoms of infection, not infected at all, or sometimes we can see hypersensitive response when there are only red dots here on the middle and actually nothing else. We don't see yellow origina, and we don't see that all the foliage is covered in origina and some tips with dieback or whatever. This resistance can be genetic, of course, but it also can be introduced by endophytic bacteria and fungi which reside within the tissues of the plants, or can be on the foliage as well. This microbiome can be vertically transmitted, or it can be microbiome gained from the environment. Speaking about vertical transmission, we can probably speculate that yes, seedlings can vertically transmitted have vertically transmitted microbiome which protect them against the infection, because we sometimes observe in the city that seedlings are growing in more or less healthy state, being very close to heavily infected hedges of syzygium. So we can see yellow syzygium hedges and near more or less fine Metrosideros excelsa seedling. So probably they have something which can protect them. Trying to investigate what is vertically transmitted from parental trees, healthy trees, to seeds and related to seedlings, we collected flower buds and seed capsules from 6 locations, 3 in Auckland City, inner city, and 3 regional parks north of the Auckland City. In each location, we chose 5 healthy-looking trees, and from each tree, 30 flower buds and 30 ready-to-open seed capsules were collected. Our main research questions were: which bacteria and fungi are present in flower buds, seeds, and seedlings derived from each individual tree? What is the variation in microbial composition among individual trees? And which microbial tags can probably promote seedling growth and protect them against fungal pathogens, including myrtle rust? Bacteria and fungi were isolated from fresh seeds Later, seeds were germinated and seedlings grown on water agar for 5 months. DNA was extracted from fresh flower buds, seeds, and seedlings, and bacterial 16S, together with fungal 80S regions, were amplified for metabarcoding studies. What we first found from culture-based approach is that most frequently isolated fungi were quite different. Their abundance was quite different for individual trees. Here on the picture, you can see that trees are marked according to the locations. So W is Vanderholm, SH is Shakespeare, Tafaranoe, Tamaki Drive, etc. And trees 1, 2, 3, 4, 5. You can see here. In each location, we can see that individual trees accumulated quite different species composition. And I also marked other species which were not frequently isolated fungal species in yellow, and you can see that in Tafaranoe, for example, Tafaranoe 2 and Tafaranoe 3 trees, they had other species which are not present in most of the trees. So probably it was a cultivation bias because, as you know, most aggressively growing fungi can just outcompete on media more slowly growing endophytic species. However, when we turn to metabarcoding results, we can see quite similar picture. Again, here we can see metabarcoding results from flower buds, seeds, and seedlings of each individual trees 1, 2, 3, 4, 5, and locations 1 Tree Hill, Shakespeare, Tamaki Drive, Tafaranoe University, and Vanderholm. What we can see here is that seeds contain the most diverse bacterial communities, and also we can see that there are quite substantial difference between bacterial communities of each individual tree, especially for seeds. Seeds contain the most diverse bacterial communities, but only several taxa can proliferate in seedlings. So when seeds are germinating, some bacteria which were present in very low abundance in seeds, they proliferate in seedlings and take over all the tissues. When we dig deeper and go to bacterial genera detected by metabarcoding in seedlings derived from each tree, what we can see is that only 2 bacterial genera, Metallobacterium and Stenotrophomonas, were detected as vertically transmitted from parental trees. As you can see here, in blue, highlighted ISVs, so its exact sequence variants, which were the same in flower buds, seeds, and seedlings. So we can assume that probably exactly these genera were present in flower buds even before they were opening, and later passing to seeds and passing to seedlings. The most interesting part is that seedlings of each tree were grown in actinic conditions and water agar for several months, and conditions were the same for all the seedlings. However, we can see a wide variation between seedlings derived from individual trees. And we also can see that for some bacteria, for example, for Acinetobacter, the bacterium made this from flower buds to seeds. We can see that yellow is transmission from flower buds to seeds, but for example, only in Tafaranoe 3T, the bacterium was detected in seedlings as well. And for some bacterial species, we can see that they were detected only in seedlings, but not in seeds or flower buds. We can always assume that it was contamination somehow. However, I would say that it is probably cultivation bias because seeds contain the most diverse microbial communities, and sometimes in very diverse microbial communities, taxa which are present in lower abundance, they cannot be amplified well, and they just don't have good results in metabarcoding. but when they pass into seedlings and proliferate in seedlings in greater abundance, they are detected much better. And also we can see here that several individual trees, they were like hyper accumulators of more diverse bacterial communities. For example, the Faranoe 5 tree here, you can see more bacterial taxa were present in these seedlings. Vanderholm tree, the same, more bacterial taxa were present, while others, they contained less diverse bacterial communities. And actually the same pattern we can see for fungi. Again, on the left, we can see number of ISVs detected in flower buds, seeds, and seedlings of each tree. And on the right, their relative abundance. And we can see that despite high diversity, fungal diversity in seeds, only few of them were passing to seedlings. And actually in seedlings, only few taxa were present. If you are going to fungal genera detected by metabarcoding, we can see quite surprising results because Acanthomicetes was detected only in seedlings and it was everywhere in high abundance. This fungus was never present in any results from Metrosideros excelsa in the past, and it was a great surprise that we can see this in actually all the seedlings except the Faranoe tree. This needs further investigations because I would not say that the Acanthomicetes was contamination from the lab. We check it down at the species level. It's Acanthomicetes muscarinus. It is a known seed-borne fungus. And yes, probably it was present in seeds, just in lower abundance, and it was not detected. It is not a fungus which is present in normal dust in the environment. So this is quite interesting. Also, it is entomopathogenic fungus. It can be detected in seeds. It is growing as endophytic in some herbaceous plants, and it also can kill insects. So it is already used to protect some herbaceous plants of commercial importance against insects because it will kill insect which are eating or sucking the plant with acanthomices. Also, we can see that most of fungi are were not present in flower buds and were detected mostly in seeds and passing to seedlings. And one of the most abundant in seedlings, which was not somehow detected in most of the trees, was penicillium, which is definitely a bias of metabarcoding, because if we are moving to culture-based results back, we can see that most frequently isolated genera of fungi were exactly Aureobasidium, Cladosporium, Penicillium, and Pestalotiopsis. Penicillium was isolated from 70% of all seeds. And again, when we're going back, we can see that only in few trees it was detected by metabarcoding in seeds, and mostly it was detected in seedlings. Also quite interesting that we can see Neophysicoccum passing to seedlings. Neophysicoccum is usually considered not seed-borne fungus and not seed-borne pathogen. However, here we can see that it was the same ISV in flower buds and seeds of at least 2 trees. And in one case, it was passing to seedlings as well, but seedlings somehow did not die. We will come back to this question later, why they did not die because of Neophysicoccum infection. It is usually killing seedlings very well, just in few days. Okay, speaking about functional properties of isolated species of bacteria and fungi, we found that some of them can be beneficial for seedling survival and seedling development. At least 14 bacterial isolates were identified by literature records as putative beneficial endophytes. 9 fungal taxa have been effectively applied as biocontrol agents. 4 bacterial isolates are known phytopathogens. and at least 3 species of fungi can be seed-borne pathogens. So here's the question: they will kill seeds or not? When we germinated seeds and recorded seedling survival, we could see that Vanderholm 1, all the seeds actually died because of Pōhutukawa. Seedlings succumbed. They were trying to germinate, but they succumbed very frequently, very quickly. At the same time, we can see that several trees, they had very high germination rate. Seeds germinated with 100% rate, and other trees, they contained Ropsia andropogonis, Culitutrichum fiorini, no Pōhutukawa in their seeds, and a lot of seedlings were lost. So now, when we speak about nursery conditions, plant nurseries usually mix seeds from different individual trees. And if you can see here, some individual trees, they contain mostly beneficial bacteria and fungi, and some trees accumulated a lot of pathogenic taxa. Let's imagine that we mixed seeds from trees with a lot of pathogenic taxa and seeds with trees with only beneficial or mostly beneficial bacteria and fungi in their tissues. What we will have? Will these pathogens jump on the seeds with beneficial microbes or not? Also, there is another question. We found that one penicillium species, probably it's even novel species from Vanderholm 5, initiated callus formation and root growth from the leaves of the seedlings. I would highlight that it is a very unusual feature for Myrtaceae in general to produce any roots from detached leaves. However, here you can see the evidence that seedling cuttings developed roots and plate containing the fungus, and leaves detached from the seedlings also developed roots. Even I just rooted some of these leaves. They were grown after that in coir pit for several months, and they were still fine, only they were not producing, unfortunately, shoots. This penicillium was also isolated with Penibacillus sp. And this is connection, I guess it's not something unusual because penicillium is associated with Penibacillus. In some studies, it was already shown that Penibacillus even can move to Penicillium hyphae and slide around them. So we have beneficial fungi and bacteria, we have detrimental fungi and bacteria. What's next? Using dual culture assay, we can evaluate properties of some bacteria and fungi derived from seeds of Metrosideros excelsa. In this case, we can just plate there in different compositions. On picture A, you can see a plate with fungus in the center and bacteria around just on the second day after inoculation, and BCDE, it's results of this dual culture assay. 14 bacterial isolates and 12 frequently isolated fungal taxa were studied. We found that Bacillus, Peribacillus, Priestia, they were the most strong antagonist of all fungi, including known phytopathogens. For example, on D, you can see penicillium in the center, fully suppressed by Bacillus amyloidoliquefaciens. On C, Aureobasidium suppressed by Pantoia. And on E, Alternaria trinata. On right, it's a control. On the left, it is suppressed by Bacillus. But also some species of bacteria could facilitate fungal growth, which is quite interesting. You can see if isolates of bacteria facilitate growth of potentially beneficial fungi, it can be good outcome for seedlings. However, if they facilitate growth of potentially pathogenic species, like Alternaria culitutrichum, it will be not so good outcome for seedling growth and their development. We also tested originospore germination in presence of bacteria. On the left upper picture, you can see how originospores germinated on water agar, and below it is Bacillus sp isolated from the University 5 tree, which was the most potent antagonist of Austropuccinia psidii in our research. You can see that most of originospores could not germinate well, and we can probably speculate that if this bacillus is present in seedlings in great amounts, this bacillus will probably suppress germination of originospores and proliferation of myrtle disease. Finally, we try to inoculate seeds derived from only one tree with 9 bacterial isolates in single and in consortium. And one seed group was also inoculated with Penicillium sp, which I demonstrated below, which somehow caused root growth and root development from even detached leaves. I should highlight that seeds were treated with antibiotics and fungicides before the experiment, and after that they were grown in identical conditions in coir peat mixture, coir peat pumice mixture for several months. Here you can see results, they are quite evident, even without statistics, but they are also statistically significant. It's seedlings grown just in water control, so they were not treated with antibiotics, fungicides, they were just as they were. B, seedlings grown after treatment with antibiotics and fungicides, you can see that their shoots are shorter, their root structure is not so well developed as for other seedlings. And actually CDE, it's a seedlings inoculated with Penicillium, Cocuria, and Pantoia correspondingly, and they developed better root structure. Their shoots are much longer, much larger. The most interesting thing is that all the seedlings derived from only one tree. However, as we can see here, we actually developed different morphotypes, just inoculating different bacteria or fungi in the seeds. Because, for example, seedlings inoculated with Penicillium on C, they all had only one stem and, okay, good root development. D, we can see that many seedlings, they developed already branches, and sometimes, as we can see on E, seedlings even developed 2-3 stems, so many stems from apical meristem. Here we can think about trees which we can always see in the environment, in the city, around the city, that sometimes we see just one trunk of pōhutukawa and beautiful crown. And sometimes we can see multi-branched, multi-trunk trees. Why they are multi-trunk, multi-branched? Because of specific endophytic bacteria in them, because of the environment, it's a big question. However, here you can definitely see that some endophytic species can enhance seedling growth and seedling development. And from our previous research, we can see that they can also protect against fungal pathogens. So we can just accumulate this knowledge and use it in plant nursery or in the field even, because bacteria and fungi are ready to grow, and probably they can be also transmitted from pōhutukawa to other hosts which are more susceptible to myrtle rust disease than Metrosideros excelsa. In summary, we can say that individual trees in one location are definitely accumulating substantially different microbial communities in their seeds. Seedlings derived from seeds of each tree retain difference in microbial species composition even after being grown for several months in identical conditions. This difference probably can influence the survival rates of the seedlings and be responsible for the variation in pathogen resistance. Our experiments demonstrated that certain seed-borne microbial taxa can suppress originospore germination in vitro, and significantly enhance seedling development. I should also thank everyone who helped, advised, and supported during this PhD, especially my great supervisors, Bruce Burns, Mahajabin Padamsi, and Haley Ridgway, Rene Johansson, who was project manager for Beyond Metrosideros from the beginning, and teams at Manaki Fennell and Care Research Auckland and Lincoln. Thank you, everyone. And yeah, I'm ready for your questions.
Thank you, Vlad. That was really fun. I love your photos, especially. It's just really neat to see the very clear differences in how the fungi and the bacteria interact with each other on the plate, and then also the differences in seedling development from those early stages. I'm happy to field questions from the audience. Audience, if you can just pop those up in the chat. I'll start with a question. Have you considered other possibilities for differences in the germination rates?
Yes, yes. Because you can see we were collecting seeds from ready to open seed capsules. So there can be possibility that some seeds were not ripe enough. And probably because they were unripe, we had different germination rate. You can see it's quite difficult to collect seed capsules of pōhutukawa because they are not changing the color before they open. And when they open, seeds will be already contaminated from the environment by different fungi and bacteria which are present in the dust. So as a result, what we can see that we need still closed seed capsules, and inside seeds will be intact. If seeds are intact but they are still not ripe enough, there will be slower germination rate or they will not germinate.
That's really tricky because those capsules are so small and the seeds themselves are so small. So I can imagine that's a tricky thing to accomplish. Yeah. We have another question here. Do you think that what you have found might be typical of other Myrtaceae?
I would say yes and no. You can see there are several different genetic lineages of Myrtaceae. And if we speak, for example, about Lophomertus and its high susceptibility to myrtle rust, it's probably because it's the same Myrtaea tribe, which is the original host of myrtle rust in Brazil. In Brazil, there are only Myrtaea tribe with soft roots is present, and myrtle rust already evolved in this tribe. So when myrtle rust arrived in New Zealand, the fungus just found the most close relative of its original host. It was Lophomertus. And it is probably why we can see that so many Lophomertus plants are infected. However, Lophomertus was not evolving together with myrtle rust. And it is probably, it just doesn't contain some microbes which are present in Brazil, because in Brazil, scientists already found some mycoparasites which eating myrtle rust. They really parasite spores and growing hyphae of Austropuccinia psidii.
Interesting.
So yeah, it can be yes and no, depending on the host, depending on the species.
All right. Well, that is all the time we have for questions today. Thank you so much, Vlad, for your presentation. A video of today's webinar will be made available on the Beyond Myrtle Rust YouTube channel in the next couple of days, where you can watch all of our previous webinars. It will also be emailed to everyone who registered. Our next speaker will be on March 19th. We'll be hearing from Rob Beresford, Principal Scientist, Plant and Food Research, about some of his recent work on myrtle rust. We'll also be making a very special announcement about an event coming up in June. Stay tuned and keep a lookout for the invite to Rob's webinar in early March. Thanks, everyone. Harira, and see you next time.